students out doing so well. I'm particularly glad to come here because I have several good friends, and I had the good fortune of meeting your president, Satish Tripathi, this morning. And I think, of course, I realized afterwards that he's an alum of the department, but he's also, he came across as a great fan. So I think you're in great hands. I'll just give you a little anecdote that'll tell you how I got into this space. You know, this is not really, this wasn't my area 10 years ago. But it so happened, in fact, not long after Champaign was a postdoc in our lab, that we had a visit from colleagues at Mayo Clinic, and they they talked to several of us, and they were very keen that somebody should go over to their lab. And they didn't really care whether we had any biology or they just wanted engineers to be there, or computer engineers and computer scientists. So it so happened that the guy who was really the chief, something like the deputy head of Mayo, I was traveling back with him, and I was going to Chicago somewhere else, and he was going, so we traveled to Chicago together. And I said to him, I said, look, I mean, you're Mayo Clinic, you're #1 in this area. We don't even have a hospital. We didn't have a hospital at the time. Why are you want to work with us? You could go and work with Harvard or Yale or Duke or any of these great medical schools. He looked at me very strangely and he said, oh no, they are our competitors. We want engineers. You can be our friends. You know, your student says what we want. And so I think it really made sense that yes, we, computer engineers and scientists, are needed by the medical guys. And so I just had a student who came in, very good analytical experience, and I said to him, look, we are going to be working in the area of genomics, because it's a computer science, computer engineering problem, pattern recognition. If you could go to Mayo for the summer, and he had at the time an offer from, I think, Cisco and some really, the Bay Area companies that were going to pay him really well. So I said, look, you may not get a great salary. I can't promise you that you'll come back and you'll have a great result. But it might be interesting. This is a risk you could take. And and Arjun decided with me to take that risk. He said, yeah, I'll go there. He said, what's the worst that can happen? I said, worst that can happen is you'll get to know some doctors. Maybe if you're sick, you can always go to Mayo Clinic. And he went there, and indeed, the work that he did there while he was there was to address this question that they had this trial that they had run, which is called the beauty trial. And it was to do with this rather ferocious type of breast cancer, which really had, I mean, the chance that somebody would live with this cancer is still very poor. And what they discovered is, not at Mayo, but I think they found in New Zealand that women who had diabetes and had breast cancer, and for diabetes they took this drug called metformin, which really was out of patent, very inexpensive drug, and their tumor actually decreased. And even now it's a bit of a mystery, but what Mayo did was invest its own money to create two sets of cells. One set of cells which were these triple negative breast cancer. And triple negative means they all these three types of estrogen receptors don't work. So it's got three negative shots against it. So they created one set of cells which were treated with this, which had this cancer, and another set of cells which were treated with metformin. And what they really were looking to see is which gene is responsible. Is there a single gene? Are there multiple sets of genes? And Arjun just had taken a bioinformatics course and nothing else. So he went, and of course he's very good with analytics. What he did, which typically people wouldn't do, we used a unsupervised learning method. But in this, there were most of the genes sort of clustered in small groups. But there were a few genes, a small set of cells, and the corresponding genes that didn't cluster, that sort of stood by themselves on the side. And they were, if you like, an outlier. And on a good day or on a bad day, you would take those so-called outliers, put them with the other clusters, and be done. And indeed, probably the statisticians at Mayo who went before us did exactly that, because they had not found anything in this to differentiate. But fortunately, Arjun did not throw out the outliers, or Arjun did not mix the outliers with the rest. And he went and said to Dr. Venchelbaum, who's also another of our collaborators, hey, look, you know, this is what I found. And what Dick did was he wasn't concentrating on these clusters that we thought were really important. He concentrated on these few outliers, and he ran to his colleague, Liwei Wang, and he said, I think we may have an answer. And the answer was that in this small cluster were a few genes that they couldn't account for to be implicated in diabetes. They looked at these genes and they said, look, these three genes we know are responsible and they really are implicated. We know all about them. But these two genes out of these five, we don't know. So Liwei could go and tell our postdocs, you've got some real homework for the next 5 weeks or so. Go and test out these genes. And of those, one of them turned out, when you sort of, this is the only time I've been inside her lab, because she's got this thing that says nuclear radiation, be careful. So I don't want to go in there. But I went there, you could sort of see that those cells that were treated with metformin and this particular, they were not replicating. Because in the lab, what they did, they forced this particular gene to stop expressing. And when this gene stopped expressing, meaning it stopped working, if you like, the cells stopped replicating, and that's the picture they showed me. And you know, Arjun came back. In fact, just this January, he joined Mayo as a faculty member. So now he's on the other side. But indeed, you know, we all need a lucky break. Perhaps that was our, if we had not had that lucky break, I probably would not be giving this talk. But the outgrowth of that was, you know, they said, look, you've had this success. We have a tougher disease for you to deal with, which is mental health. And in mental health, of course, there is no, the ground floor is very low, in the sense that all you have is the doctor, the psychiatrist coming and asking 20 questions, and based on those 20 questions, and they tell you what drug you should take and so on. But it's it sort of gives you a sense of what we as computer engineers and scientists really, how we can make a difference. And increasingly I see lots and lots of these opportunities. And from that one, this is Arjun here, and of course these others are people who got equally excited. And I think the most excited people I see in my lab are not even my graduate students, but the undergraduates who come in, and engineers, electrical engineers, computer scientists, who think they can really change the world because they can have an impact. And that, I think, I find to be very exciting. These are some of our Mayo colleagues. Unfortunately, the line keeps growing because, and I started by telling you my experience with Mayo. I mean, it could have been the same experience coming and working at at your medical school, but the good thing about, I think, the Mayo Clinic was and continues to be is that they do recognize the opportunities and the need for these kinds of partnerships. Out of that kind of the work that we did, we now have a more formal alliance with Mayo in that our students now, take the pathway that Arjun and others treaded and go and spend the summer on salaries that are so much lower than what we can get you can get by going to the Bay Area and working in a Bay Area company. But they come back, they are, bright-eyed and ready to go. So this work promotes a broad spectrum of collaborative research. By and large, while it's not only restricted to engineering, it's substantially, it is between engineering at Illinois and the Center for Individualized Medicine at Mayo. We are focused on developing new technologies, new ideas that come about when we are there and our students are there. There's also a strong interest in the new generation of medical students and early residents who are coming up to learn about what we do. In fact, we had a whole bunch of Mayo students just this past weekend, spending the time trying to go through classes, and it's a tough time for them. There are some things that actually allow us to work together. I think NSF has funded a center for computational medicine at Illinois. I mean, it's jointly funded between Mayo and Illinois. And of course, with those small projects, we now have increasing number of faculty, both from electrical engineering, computer science, mechanical engineering, participating in this with Mayo Funds fellowships, very competitive, some grand challenge projects, and I think the summer internships probably are probably the most popular because they achieve a lot. And in fact, this is my one slide that says how, and it's a slide that Arjun gave me, and you can sort of see what happened here is that there were 3 of these clusters that had most of the data points, and a tiny one here that span across that had 230 genes to start with, which boiled down in the end to the single gene, which is CDC42, which really started to reveal the pathway that now I think at least they know that there is one gene that is responsible. If it is downregulated, it stops the tumor from spreading. However, that doesn't say there are not other genes that are equally or even more responsible. But at least I think we started the question being asked. So what that essentially said is that this joint work can really make a difference. We then got, I think, as I said, a big factor in how we work together is CCBGM. My co-conspirator there is Dr. Liwei Wong, who's an MD PhD with a focus in pharmacology and medicine. And as you can see, the first problem she gave us to do was, in fact, to look at drugs and the efficacy of drugs. And that continues indeed to be a major thrust of our work. So what are the things that we do? The vision of the center that we have, as well as, I think, the strong interest and the expertise that we bring at the core is really how we manage data, how we manage different types of data. An important aspect of working in the genomic space, in the broad omics space, metabolomics and using epigenome and all this, is that you can't simply go and say, I've got 4 varieties of data, I'm going to just normalize it, standardize each one statistically, and put them together, and yes, I have an answer. Because it's really the semantics of the data, what the meaning of the data is, has to be preserved. Otherwise, it's garbage in and garbage out. So an important aspect of what we do is indeed manage the data. The data is not always large. It is complex. Patient trials, particularly, I think, may have 1,000 patients, 2,000 patients. That's perhaps a significant trial. So you can imagine this is not the Google kind of data that immediately you run to a TensorFlow engine and get your result, or train it and get results. It really requires quite a bit of thought, how you put the data together, and then the kinds of methods we use really are what I'd call these probabilistic graph models. Those of you who have some experience with probability, have some experience with conditional probabilities, Bayes' rule. I mean, in the final analysis, that's the core of a lot of the models that we use. It at once, I tell my students, all you need to do, most of this mathematics is your high school calculus and a little bit of probability to start with. And it's truly the combination of that that drives the analytical tools. We also, once we have this, once we know how to manage the data, once we know how to go and do the analysis, we actually go and build systems. In fact, this is one of our significant projects, is to build an engine that can handle quite a bit of this graphical models and execute it with efficiency. So, keeping these things in mind, the key question, set of questions that we try and answer are, how do you take information, genomic information, which is not insignificant, it's quite large. Other kinds of omics information, for example, metabolomics, and if you're looking at drugs, it sort of tells you how the drug is being absorbed or utilized by your body. And of course, patient history. And then, you know, the real opportunity is you combine this and produce what I now call actionable intelligence. But I should tell you how this term came about, at least in my thinking. I was in one of my early days, I was giving a talk, I think perhaps my first talk at Mayo, and it's very early in the morning. These physicians and surgeons, they started, you know, for us, really an ungodly hour. It was 7 a.m. in the morning. They give you breakfast, and then you give the talk, because, you know, 7:30 or 8 o'clock, even earlier, they start going and operating and doing whatever their rounds and so on. So I started, you know, I thought, of course, these guys don't know too much of what I do, you know. So I said, look, we can determine from the genome, we probably have the fastest algorithm that would go and determine the mutations, the variants. Now you can use that in your work. And I might have been 10 minutes into my talk, and the surgeon raises his hand. He said, look, I'm a surgeon, and you give me 100 or 1,000 mutations, it's of no use to me. You have to tell me the one that I can act on. So it really drove home that what we think as being useful to the physicians or to the patients is really not, unless we tell them, or they can derive something actionable. Biomarker candidates, you know, patient-specific predictions and therapeutics. You know, are the drugs effective for some versus another? And a problem that really became very important, I think, in our work is really the major depressive disorders. You know, depressed patients, in fact, constitute a significant loss in society. The economic burden, the economic loss due to depression is huge. I was at this insurance company, Optum, in Minnesota, and they claim that just for that one company, the cost of treating depression is of upwards of $10 to $20 billion per year. And it's not because the drug for depression is expensive, it's in fact very cheap. The real impact is that the cost of side effects, the cost of patients having to be treated for other problems because these drugs are not working for them. The patients, sometimes they themselves self-medicate and take more than what they should. The impact on the families, all kinds of things that are related make this very expensive. So it's while psychiatry as such, in the scheme of professions, medical professions, is not the one that stands out and most attractive for people to go into. It is in fact, the psychiatric diseases, in fact, or the mental health diseases, they believe are the problems that are going to be going to really skyrocket going forward. So, just to give you a sense of what our thrust is, so our goal, as I gave in the starting example, is to see how we can individualize a treatment or a drug by a specific patient. So in the depression, you know, the faculty will appreciate it. I mean, we as faculty are always depressed, right? It's something or the other, not going well. We always look to things that are not going so well. So maybe, you know, Ken and I both are depressed. We go to our psychiatrist. Now, the psychiatrist prescribes the same drug to both of us. It turns out the drug is 80% effective for me and is only 20% effective for him.
Wow. Happened.
Sorry.
I can give an example.
Yeah. So indeed, this is true. On the average, the drugs that are prescribed to be approved by the FDA have only to be 50+% effective, sometimes maybe even less. So can we go and say, not by changing the drug chemistry, because we can't go and change it. I'll never be successful in that business. But can we take the patient's chemistry and use that to go and say how effective the drug will be for him or her? You know, that was the question. That indeed became the question. These never become, you know, these questions get defined as you go along. But in the end, that was the question that we tried to answer. And what we came back with is that yes, we could do it. And what we have to do, of course, is we're not going to replace the psychiatrist. We're not going to replace the surgeon. So I'm not there making the claim, for example, Watson does, that yes, we can replace the cancer surgeon or the oncologist. And here, what we said is we can actually provide actionable intelligence to the psychiatrist so that this an effective drug can be prescribed to the patient. It may be that the drug that we both got only works for me, but there might be another drug for Ken that is equally 90% effective for him.
I changed drugs.
I'm sorry?
I changed drugs.
You did.
So I know that, yes.
And this is feasible. I think this will happen, and this is really what we have achieved for one disease. And because of that success, I think there is, I mean, not just hope, but there's also lots of interest to work and to actually influence this field. Similarly, of course, the story, another area that we work in is in neurodegenerative diseases. Here, the problem is slightly different. You know, we've had almost all the drug trials that failed. So, yes, there's a lot of work in early prediction of Alzheimer's, of dementia. But let me pose the question about drugs in the following way. We could find a drug for effectiveness for depression. Could we take a failed trial and then, rather than go and say, this drug trial failed because clinically and statistically we couldn't find a significant group for which the drug works, and so we give up, and you know, the last drug trial that Eli Lilly had in the Alzheimer's space, I think it costs, they claim, at least $600 or $700 million. And in phase 3, where I think they go to phase 3 when as you go through various stages, it's almost worse to fail at that stage. But could we take that data? Could we take that information and determine the characteristics, the cohort for whom the drug is effective? Everybody knows. In fact, you go and ask people who run these trials, they tell you, yes, the drug, I'm sure, is effective for some. And they have a sense. But there isn't a proper method that says, as the trial goes on, imagine the trial is now set up in a different way. As the trial goes on, we learn about the characteristics of people for whom the drug is working using reinforcement learning, and for those of you who know, I mean, this is how babies learn. The baby gets up, she sees that there's a world out there, very interesting. She decides to walk because she's rewarded. So similarly, reinforcement learning rewards success. That is the direction in which you want the program or the system to learn. So could we actually, we said, use reinforcement learning together with the domain knowledge that these physicians already have. They have a sense of how effective the past drugs could have been. So if we put this together, we start to remove people for whom the drug doesn't work. It may look to be a little cruel that you take people off the trial, but you end up at the end with a cohort. Maybe it's not 55%, maybe it's only 35%, but you have the characteristics of those 35% for whom the drug really works and works well. 80% effective, 90% effective. So now, yes, Eli Lilly didn't go and win all of its $600 million, but now we have a characteristics and a cohort for whom this drug can work. Okay, so if we did that, we said yes, we can start really finding ways in which to retrieve success from failures, and start, and maybe that gives insights into my colleagues who go and work in the labs to start designing drugs that might work. I mean, I don't know how that would happen, but that's the idea. So what we have done is shown that this kind of approach works retroactively in stroke patients. I won't really go through the details of that here, but that sort of says, yes, such an approach, reinforcement learning together with, of course they are again, as I said, these are graphical models. They're not really, this is not a problem that you have enough data to feed a deep learning engine, but there is enough data to build a Bayesian model, more generally a factor graph model, which says how the patient progresses in time as the drug is delivered, and at some threshold lets you predict success. So so part of the work also is for us, as I said, to build a system that brings these together. But I'm happy to talk about that, but I will really focus more on the depressive disorder. As I said, major depressive disorder is something that affects many in society. In fact, what these are some major celebrities over the years who have had depression, and in fact, probably I think half of them are no longer alive because they committed suicide. So it is a severe disease. It's a major cause of disabilities worldwide, very complex symptoms, and unable to predict well treatment outcomes. You know, psychiatrists find it very difficult to go and say who's going to improve and who's not going to improve. And it's not the cost of the drugs really that makes a difference, it's really the side effects that makes a difference. And side effects is what drives the cost. So clearly they deserve better care. And more than that, the societal impact is quite large. Currently, if you look at the best data there is, the effectiveness of the drug is at best at 55%. That means you can luck out and become well, or you can be unfortunate and have no effect. It's slightly better than chance, slightly better than a coin toss. So, in a sense, it's a very ripe area to make a serious difference. Okay, I mean, the ground floor, if you like, is very low. So our approach there is to say we have patients who are assessed by the psychiatrist, and as I said in the beginning, this is the psychiatrist and the patients, depending on the scoring scheme that you have, score. The psychiatrist gives the patient a score. The patient gives himself a score. So you have two sets of scores, and these are questions. Are you sleeping well? Are you eating well? Did you feel bad yesterday or the last few days? And various and some fairly personal questions that come in, and based on the higher the score is, the worse you are. And what the psychiatrist does is invariably he or she will give me as a depressed patient a drug which is fairly common, and these are called SSRI drugs, and they have, of course, they work. I mean, I still don't completely understand the chemistry of these drugs, but it is their side effects have been documented reasonably well over the years. So we have data from the trials that are run for these drugs, and we had data not just from one trial, but several major trials, and some of these trials were in fact pioneered at the Mayo Clinic, so it was a good, really, way for us to start. In order to look at the progression of an individual patient through the process of taking the drug, and trying to go and say at what time can we predict how effective the drug will be, what we did was build a type of model that is called a probabilistic graph model. The simplest form of a probabilistic graph model is a Bayesian network, which of course doesn't deal with time, but sort of statically describes the progression or the impact of whatever phenomena that you are interested in. In this case, the impact on the patient of this drug. And of course, you train it, and here the training consists of getting the conditional probabilities, which is clearly it's a Bayesian network, so conditional probabilities are important. And the patient, the physicians know how, which are the priors, and then using the conditional probabilities, the starting conditional probability, and the prior information, the graph lets you then walk through and evolve the impact on the patient of the drug. And essentially, I mean, if you, in a nutshell, the difference between doing a graphical model like this and if we had a massive amounts of data, pumping this through a neural network is that in both cases, you are building mathematically joint probability distributions. Okay, so I have, and these are high dimensionality spaces, so you have to build fairly complex distributions. And essentially what you do with the domain knowledge is to say, I don't really know how to build the total joint probability distribution, but I know how to actually build pieces of it. So I can factorize it, and if each of the factors has got sufficiently small amount of dimensionality, then I can solve those, and you know, voila, I've solved the problem. You see, it's at once, you could say, the limitation, or depends on your point of view, the beauty of addressing it using graphical models. So if you have the domain knowledge, and you have information from the physician about what are the priors, or you can extract it from the data, then for a problem-specific solution, this is a great way to go. And, you know, it's not, again, one size fits all. You have to build the price you pay is that you have to build a graphical model by the problem. You have to really learn what that model is and construct it. But if you can, if you have a patient enough psychiatrist or a set of psychiatrists, if you have data, which is not usually large, then this is a great way to go. And indeed, in this case, you know, I said that our first success was looking at the effectiveness of metformin. Here as well, I mean, you could say we lucked out, in part because I was always intrigued by this method. I taught it, looked at other people's examples, and there are difficult aspects of it, but was really very keen to go and see if such a model would be effective. Well, if the model was not effective, then I wouldn't be here giving the talk. You know that. So what it really did for us is to provide clinical actionable intelligence, and it really now helped the Mayo psychiatrists improve the clinical decision making. So we didn't go and say to them, this is going to give you the answer. What we said is, this is going to help you make the decision, because it's going to tell you that for this patient, with this kind of record that you've collected, and with some other information that we need from you, you can make a real decision on how effective this drug will be, or how effective it will be based on what you're observing. And so here's where I think some more interesting aspects come in. We had, as I said, we had the scores that the psychiatrists had given. We were looking at how effective the drug will be for the patient. So what we did was, we had in fact data. I mean, this is you know, you take a blood draw, and the Mayo clinicians, they collect information on the metabolites that are really most effective in that particular individual, and the individual's genomics information. So here is just two of the pieces of information that we went with. The first says, how is the drug being metabolized in that particular patient's body? And you can start to see how this is trying to personalize it by the individual. Next, what we did was we looked at the genomics, and it turned out if you take the whole information about the person's genome, it did not really help us in improving or in saying specifics about the patient. It just got, if you like, lost in the noise, because the impact here was very much related to how the drug is getting metabolized, getting absorbed in the body. What mattered was that the genomic SNPs, or those mutations that the person had, which were related to the metabolites that were highly responsible. So when we put the data together without any of this information, the results were totally ineffective. But once we picked, and here with the help of our physician colleagues, we picked the top 5 metabolites, and then determined which metabolite was indeed stronger in that individual patient. The patient's mutations that were particularly relevant to the metabolites that we picked, and once we did that, we then, using our graph, we started to make personalized recommendations for treatment. Okay? So the result of that, and I won't go into the details of the model, but what essentially you start by breaking up the patients into clusters as to what their scores are. So the first level, the ground floor, is how the physician scores, so physician's insights. And this symptom dynamic says, how do these symptoms change? And which is really, here's where the graphical model comes in. And looking at the change in 4 weeks, we can start predicting what will happen to this patient in 8 weeks and 12 weeks and beyond. And having the information on, modulating this with this information on the metabolomics, the drug absorption rate, as well as the genomics, really serve to then go and accelerate the level of predictions. So let me, and this particular paper, in fact, is appeared in Computational Intelligence, and if you really need a link, just send me an email. But here are the 3 different trials that we went with, and in fact, that's probably all of the data that's available pretty much anywhere. The first is a trial that was pioneered by Mayo, by a very senior psychiatrist at Mayo, and you can see that the number of patients is relatively small. And part of the problem here is that people who are depressed, it's very hard to convince them to go and join a trial. It's difficult to convince them to go and see a psychiatrist, let alone stay in a trial for 8 and 12 weeks. Even if they are there, as soon as they start feeling these side effects of the drug, they drop out. So you don't end up having a strong cohort. In the beginning, you really have to persuade them to join the trial, and you have to sort of keep their spirits up to stay with it. And these trials, or these data collection process, it's not, the physicians don't get the kind of salaries we engineers get. They get maybe, I've never seen a physician not drive a very, very expensive car. So you can see why these trials are expensive, because you've got to pay the patient, the psychiatrist, a lot of money to go and participate in it. Don't tell my psychiatrist friends who said that. The other one is a National Institute of Mental Health national trial with 1,200 patients, and IPSC is an international consortium, so there were patients from Europe, patients from organizations in Japan, so it really spanned quite a number of countries. So not only are we seeing locally how things are, we're really getting as good a picture as we would ever get of how depressed people are throughout the world. You know, as much as a picture of the world that we could get. So, using the kind of the graphical model that I described, this is what the prediction rate was. So with the first, the Mayo trial, for men it was 75 to 80%. For women it is higher, so women generally scored higher in their ability to improve under treatment. Yes?
Could I just ask quickly what the outcome measurement is?
Very good question. Yeah. So the question is, what success, right?
Yes.
So there are two levels of successes that the psychiatrists agree upon. One is if you start with a symptom score, so it all starts with the score that you get in the beginning. So I go in very depressed, I may have a score of, I don't know, 50. If my score comes down by 50% as a result of the treatment, then I am supposed to be responding. If it comes down to 5 or so, to 5 to 10% of what you started with, I'm supposed to have remitted. Okay? So that's what goes in. These are remissions. Okay? We also have numbers for response, but these are remissions. And the initial 55% that I showed you, which is the norm today, or the best that they can do today, that is also for remission. So slightly better than 50% of the people remit. So at least in that period, their disease disappears. You know, they may still be struggling with side effects while they're taking the drug, but the symptoms of the disease, how they score themselves, disappears. And you can see, you know, the international trial had slightly higher numbers, but here the difference between men and women was not that great. But overall, we did concur, I mean the psychiatrists did concur that women do in fact do better than men. So, because of the success of this is of course a retrospective trial, execution of the data. What Mayo, in fact, just a week or so ago, they decided is that now they will go and invest their own funds, not wait for NIH or somebody else to fund it, to launch this into practice. Meaning they have to go through the psychiatrists, get them to start using this method, and use it, and start to see how well people improve going forward. I mean, we said, if you took the historical data, this is what your results would be. Now, how does it really work when you start using it in real life? I think a very important question. But without what we did, they couldn't do it. Now there is a tool, there is a methodology to go and make the prediction, and the psychiatrist then uses his or her own knowledge together with the predicted information, to decide how to treat the patient. And invariably, in this, there'll be two groups of people, one who actually just go and get treated the usual way, another being treated the way that our tool, in fact, augments, and then see what is the difference. So let me, and this is the interface that indeed psychiatrists are going to use. And in fact, it's already starting to be in use. And I'll tell you a secret. You know, they say students code, graduate student code, and this was put together by two of our undergraduates. They came and they were very eager, and I said, look, we have this tool. It has a terrible interface. No physician will use it unless we make it pretty. So these two students, they both, I think one is a computer scientist, another was a computer engineer. I mean, very good students. And in about 3 or so months or so, they created the first interface. And we took it to our Mayo psychiatrists. They made some suggestions, and this became the interface. So if anybody tells you that our students don't code well, don't believe it. You know, we're just as good as anybody else. And, this is not even graduate student code. This is really undergraduate student code. So where do I see opportunities? I think the opportunities in building, of course, if you are working in imaging, I think there's nothing better than deep learning today. There's nothing that will beat it. But as soon as you take the imaging information, be it in neurodegenerative diseases or in mental health where imaging is starting to come in vogue, if you need to integrate it, then I think look for models that are specific to your problem. And I really think that a combination of graphical models and, of course, simple analytics can make a big difference. An area that we are exploring going forward is to bring in the Bayesian networks that we have been playing with, the graphical models, together with deep learning, because between them, I feel you can not only use domain knowledge to help determine what the right weights and inputs to deep learning engines would be, but you can advance the interpretability of your deep learning answers, if you like, a whole lot more. And we've been experimenting with it. So you start with the kind of information that we started with to build a graphical model. You then use a small, not such a deep engine, but a small neural network to make a rapid decision on what the outcome should be. So real building from information to cognition, combining these two ideas is, I feel, a very, very important area for us to pursue. There's already starting to be some valuable theoretical developments in that area. But coming to, I think global wellness, it's the figure that I see from WHO is something like US 16 trillion. It's a big economic burden. Patient-centric design, patient-centric medicine, individualized, be it in cancer, and in fact this is something that as working with those work in liver cancer. My colleague who's a Mayo physician, he's led this in cancer therapeutics, trying to say, given a particular type of cancer, for whom will chemotherapy and other drugs actually work, and for whom it will not work, and actually provide patient-specific treatment, but also while reducing side effects. Neurodegenerative diseases, mental health, I think are three very, very important areas. Thank you very much, and open for questions.
Okay, great. We definitely have some time for questions.
Yeah.
So when you look at some of the recent studies on mental health, especially when they talk about healthcare delivery for patients with mental health, they always talk about looking at environmental factors, community support, lack or presence of community support, socioeconomic background, and so on. So are those things built into this framework? Maybe the initial symptom score, does that include that, or are there plans to integrate those aspects as well?
It's a good question. The symptom scores really are very personalized. They ask, how are you feeling? I mean, how are you eating? How are you sleeping? I mean, all these. They don't ask you. There may be a couple of questions depending on the kind of scores, whether there is violence in the house and things like that. Some domestic aspects. But do you live in Beijing or New Delhi, or do you live in Colorado? No, they don't ask, and that is a big impact. In fact, in this current cancer study, that is an important factor. So it comes under what you're exposed to is called the exposome. What kinds of chemicals they find in your body. Methylation. How various methyls are being expressed related to gene expressions. So these, in fact, from what I understand, when you take this whole lot into account, the impact of the genome in and of itself is perhaps 20%. It's the remaining 70 to 80% is metabolites, it's all kinds of other things that are going on. So yes, the genome is an important aspect of the story, but it's one of many. And its total impact is still not quite understood. Because even now when we see sequences, there are many, many gray and dark areas that are not used at all. So maybe when they all come to light, things will change. Yeah.
Question?
So when you're talking about your graphical model, obviously your goal is to predict the drug which would be most effective for the patient given their particular circumstances and their biology. Is that done in kind of iterative fashion where you assign them a drug and see how they do, and then you, based on that prediction, you assign another drug? Or is it done, or is that AUC calculated by basically assigning them one drug and seeing how they do in a single?
Okay, so let me back up. I mean, you really need data for this.
Yes.
Right?
Obviously.
And from the data, we derive a. I don't know how familiar you are with building the graphical model, but I mean, if you're using, say, HMM or factor graphs, you need to determine the relationships between states and observe. You're making an observation. There are these hidden or unknown states. You're trying to determine what the state that the patient is in during the course of this of the drug being given. And what you train this with is previous information on how patients have reacted.
Yes.
And, you know, I mean, we did it for one drug, but you can do it, now we have done it for 2 and 3. So you can have a graph that sort of is an accumulation of multiple drugs.
Okay, so you were selecting patients that the drug would work for, not?
I'm sorry?
So I guess I was confused. You were selecting, you had one drug and you were selecting a cohort of patients that.
For whom
that
the drug is
would
effective
work
or
for.
not.
Okay.
Yes,
You weren't
yes.
assigning drugs to patients.
We're not, I mean, so far, we're not sort of assigning drugs. In any event, what you may have to do is that with multiple drugs, you may have to start the patient on one drug. If it doesn't work, you find out very quickly.
Yep.
An alternative is that without really going through, you can sort of say, what's the likelihood, given the metabolomics and the genomics of this individual, that the drug will be effective for that individual? That is an initial starting point.
Yep.
Using that, you say, I'm going to put you into this stream, and it's possible that your initial priors were not so good, so you still have a story to tell to this patient. So you derive the probabilities from the data, and essentially you're designing the factor function, which in fact is the key, right? All of the mathematics is in the factor functions.
Yep.
And so we construct the factor functions, derive the parameters from the data, and now using that, by patients, we dynamically construct the graph. Okay, so it's not the usual factor graph where the whole thing is done. All of the parameters are there, but I come in, I have a graph, I have a set of states, and depending on those states, success or not depends on whether I exceed a certain threshold.
Yep. So
You know?
in the future, will you, let's say you assign, or you determine that this drug will work for this patient, and it doesn't happen, will you use that as a prior? Or-
That goes back into the data. All the failed information goes back
Yep.
as a, you know, continuing to learn. So, yeah, so I think, but the good thing is that we don't have a huge amount of data. And this kind of a model
We're
where,
both from the pathology department doing machine learning there. so
Yeah, yeah,
We know
yeah.
those issues.
so you don't have a lot of data, but
Yep.
Pathology perhaps you do, I don't know. You have images,
Depends on
right?
you have a lot of images beyond a lot
Yeah,
of annotations.
yeah, yeah. Yeah.
Sir, we like right now the needs are by the physicians and.
I'm sorry?
The needs currently are by the physicians, and computer technologists and engineers work with the needs which are risen by physicians. From your experience, do you think that needs would be generated by computer technologists, which physicians would bother to carry out with the cycle reverse? From your experience, could you comment?
That's a very good question. You know, I always think that I have the great ideas, and my job is to persuade them. Are you a physician?
No.
Okay, so I'm not offending anybody here. My job is to persuade them to use these methods. Actually, my Bill Bobo, who's a head psychiatrist, I mean, he just has a passing knowledge of what's a factor graph. I could never sit down, just like he can never sit down and explain to me how he makes his therapeutic decisions. I could never completely explain to him how this whole thing works. So so who is educating whom, I don't know. I think in the end, he becomes a little bit of a AI person, enough that he's dangerous and he can give a talk to other psychiatrists without me being there. And I can come and give these kinds of talks where I don't know enough about any of these drugs, but I can still give a talk and write papers. So we do publish in computing conferences indeed. I mean, my students have to, otherwise they won't graduate. And of course, Bill helps us in publishing medical journals, where, I mean, the culture is very different. They have many, many, many more authors than we do in our papers. So the answer is yes, we educate each other, really.
Yep. Question here?
Yeah, again, from the computer science point of view, do you incorporate self-reporting of data, especially with all these computing gizmos that you have, watches and things like that available, especially in the case of depression? You get fine granular data if you allow self-reporting, the same kind of interface that you give for the physician to the patients, and maybe that will be more frank, or it's become methodical, routine, or something like that. And again,
so yeah, I mean, in this case, we have two sets of reports. The physician's assessment. I am a person feeling depressed. I go to the physician. They give us both forms. I fill out the form. She fills out the form, and to be honest with you, they rely much more on their forms than they do on mine. But what we did was to look at both. There are significant differences. So we pulled out 4 characteristics that they both are closest on in doing our analysis. But if you sit down with the psychiatrist, he will say, I truly go by what the patient says, but actually when you look at the report, because the psychiatrist seeing me for an hour, I'm living with myself. So that they believe their own numbers better than they believe ours. That's the nature of the beast. But hopefully, analysis like this, which shows that where the symptoms agree, is where we have the biggest success. And it is making a change. We presented this work at a very major psychiatrist conference. Huge interest, yeah.
Okay, other question?
Yeah.
There we go. Okay.
Earlier you mentioned that the data which we get, the biomedical text which we have, we cannot plug it directly to the model. So like what kind of pre-processing will you go through? Because like currently I am trying to pre-process some amount of data with these drug-drug interaction, and it's just like impossible. I just feel like it's pretty, there are many cases, and once you try to solve an issue related to particular condition, you end up uncovering multiple issues to
Yes,
particular
yes,
information.
yes. So we have an algorithm that uses the semantics. I mean, what you're saying and what we observe also. It's not just the statistics of the data. It is the semantics. It's what the data means that must be included as you start fusing the data. So we start with the psychiatrist's evaluation, which is a human assessment, which is a number, score. And on that score, we say we are going to map the top metabolites that we've seen make a change for this patient. So that's extracting the meaning of the metabolites. Then we say, we're not going to have all of the genomic information, but only that part of the genome where the mutations are that are closely related chemically with this metabolite. You see, so we have a stack, but that's a semantic stack, not a statistical stack. And I think unless you do it, the answers will just be, I mean, they won't make any sense. Unfortunately, people don't do that, because I think if you are, where a lot of this data fusion has come from is from the sensor world. There, I think I can, if I'm taking wind and so on, and or an airplane or what have you, it's okay to put, to characterize all of them in a normalized scale. Here it's not. Yeah. Does that help?
Yeah, and also like, have you considered using the linguistics information about the reports that the psychiatrists generate, and they write down a lot of notes? Have you like considered about utilizing those by like applying some computational linguistic models?
Very good question. So far, we haven't. So far, we've just taken the numerical scores, and our predictions are good. But you know, there is information in the, there is. We have some, I had a student who actually went and extracted some of that information. We haven't done it yet. Yeah, good point.
Okay, is there any other question? All right, take one more question. Okay.
So, I don't know that much about medicine. I'll preface my question with that.
I don't either.
So, the idea is that for each patient, the curve with which medicine affects depression would be different. So, has it ever happened that you set a threshold that after this we kick him off the medicine because it's not working? And so so it might be that it might kick in at a later stage.
I'm sorry?
It might kick in at a later stage. So in the cycle. So how do you account for something like that where the medicine might not kick in the stage that you're observing, but it might kick in at a later stage, but he's off this medicine anyway and it does not help?
I think it's possible. But you know, here the question is if the medicine is not working, or at least we are predicting, you know, I mean, I can't, we're predicting that this medicine is not working. We are sort of, there is a chance that you sort of give them more, it works. And we have that data. We have data on how much they have given to a particular individual before they gave up, and what was the trajectory. So we have that. So it implicitly takes into account that there is a potential for a change to happen. But I think in our model, that impact is less. We don't really weight it that much, because that's where domain knowledge comes in. The psychiatrist doesn't weight it very much. So it may be the trajectory did go like this. And if I emphasize the apps, I mean, they go and say, well, you know, on the average, the trajectory is actually going down. So this patient is going to have increased side effects and reduced impact of the drug. So let's sort of take him off the drug. Okay, so the machine is actually learning from what the psychiatrist has done in the past. And in this graphical model, it sort of says, at this point, I am convinced that the drug will not be effective. And convinced would mean some threshold. So there's a decision-making logic that kicks in and says, have I exceeded that threshold? How confident I am that I've exceeded the threshold? And then it gives up. So not real good answer to your question, but you know, that's where it is.
Okay, well, let us all thank. This is a great question and answer session, I must say. And after this, we would have to pay him overtime. There are light refreshments over in that corner. Thank you very much to you all for coming.